get going because I know everyone's feeling tired. I certainly am. This is going to be a bit different because I'm a cell biologist and a biochemist. So I'm going to be presenting cell biology, but I really want your input in terms of thinking about how this could be modelled or use biophysics on it. So, and I decided to really focus today on something that has come up through other talks, and that is how cells deform to get across. This morning you heard about how cells deform to go through extracellular matrix. I'm going to be telling you about cells crossing another cell type. So, in principle, it's the same idea. So I'm going to be talking about transendothelial migration, so how leukocytes and cancer cells cross the endothelium that lines blood vessels. And so if you think about a blood vessel, I mean, I haven't drawn this in 3D, but in vivo, the cells there's fluid flow, and the leukocytes or cancer cells, they have to do 3 things. So first of all, they have to attach. So there needs to be some way for them to attach to the endothelial cells. The second thing is that they have to cross this endothelium. And there are 2 ways to do this. So one is to disrupt the junctions and squeeze between 2 cells, because these cells are attached to each other through lots of molecules between the cells. Or they can go through the endothelial cell. So that's called transcellular migration. I'll show you a movie in a minute to show that. And then once they've gone through, then they have to pass through what I've drawn here. This is the basal lamina, which the endothelial cells secrete. It's extracellular matrix. And they have to cross that, and then they have to move through the tissues, which again is extracellular matrix, to get to where they're going. Now, if you think about this, a leukocyte does this all the time. So leukocytes are designed to move, and they need to cross the endothelium to get into tissues, for example, during inflammation. So if you have a wound and you want the leukocytes to fight the bacteria that got in, then they have to get up. So a leukocyte, its goal in life is to get into the tissues where it's going to migrate to wherever it has to get to fight the infection. But cancer cells are not designed to be inside blood vessels. So leukocytes are designed, and cancer cells get in there by mistake. They're not people, they haven't got brains, they just do it because it happens. So they're growing out in the tissue, so I'm thinking really mostly about epithelial cancers here. And they become invasive, and they move towards blood vessels, sometimes towards chemoattractants that are presented around blood vessels. And so first of all, they need to do the opposite to what I told you a leukocyte does. They cross the basal lamina, which is extracellular matrix, and then they squeeze into the blood vessels, and then they circulate. Now, fortunately, most of them die because a lot of cancer patients often have circulating tumour cells in the bloodstream, but it's very rare that they get metastases. And the reason for that is cancer cells are not designed to be in the blood vessels or to attach to the blood vessel wall. So it's only leukocytes that are designed to do that. So very occasionally, cancer cells acquire adhesion receptors which allow them to attach to the blood vessels. But that's not a normal thing for a cancer cell to do. So the endothelial cells are designed to attach leukocytes, not cancer cells. But occasionally cancer cells will be expressing the right adhesion molecules to be able to attach to the blood vessel. And once they do this, then they can, theoretically, they could just sit there and stay attached and proliferate in the blood vessel, or they can cross and go out into the tissues. Okay. So this is a movie, so I'm going to contrast these 2 processes. So these are leukocytes, monocytes, which are normally found in the bloodstream, and we're adding them to endothelial cells, which are HUVECs, which you've heard about before from previous people. And so the monocytes attach, and then you can see that they very rapidly cross the endothelial cells. So these are the monocytes, they're round and bright and these are the endothelial cells here, and it's a confluent monolayer. They're all attached to each other through cell-cell junctions. So in the movie, what you see is that I'll show you several different cells. So this cell here is exploring on the surface, and it goes between two cell-cell junctions, and it's moving underneath. Now these cells are trapped because it's on glass, so they can't go anywhere else. But the important thing is that leukocytes can deform their nucleus much better than cancer cells, so they can move underneath the endothelial cells which are on the glass. So they move really efficiently under there. So you'll see that again, but I'm going to put your eye on a different cell, which is this one here. If you just watch that, cell in the middle, this one, it goes through the endothelial cells. So instead of going between two endothelial cells, it actually deforms, makes a hole through the endothelial cell and goes underneath. So it's not going through two junctions. What's happening is the two membranes of the endothelial cell fuse. There's a pore, it goes through that, and then they refuse. So it's no more difficult than going through a junction, really, for a cell. Okay, so they do it really quickly. So they cross the endothelium in about a minute, from top to bottom, or even less. But cancer cells don't do that. They, for a start, they can't form the nucleus, so they can't get underneath the endothelial cells in this same situation, exactly the same, except we've labeled the cancer cells with fluorescent proteins just so they're easier to see. So blue fluorescent CFP, which looks blue, and YFP, which looks green in these movies. So it's the same thing, except this is a 6-hour movie. And the cancer cells do something completely different to the leukocytes. So, here we are.
No.
Oh, sorry. What happened? Oh, I pressed this thing. Okay. So what they do is they flatten out and make a hole between the endothelial cells. So they push the endothelial cells apart, and then they flatten out on the substratum, and then they move around with the endothelial cells. So they're actually sitting in the endothelial monolayer in this case, whereas the leukocytes squeeze straight through and went underneath. And this is a slow process. So they can attach, these particular cancer cells, we've selected them because they can actually attach to the endothelial cells, and many cancer cells can't. But this particular cell type, the cell line we use, can. But they attach initially, and then they spread and sit in the extracellular matrix with the endothelial cells. So, but they can actually cross if we give them the right environment to do that. So if we plate the endothelial cells on a thick layer of collagen, and then we add the cancer cells on the top of the endothelial cells, just as you saw in the movies. But in this case, we've got collagen gel underneath, so the cancer cells theoretically can go into this and then get away from the endothelial cells. And that's what we see. So this is an XY, and this is looking in the Z across here. So you can see the endothelial cells are red. They're not really red, but you know we've labelled them with the red dye. And the cancer cells are green, and you can see that at early time points they're just attached on the surface. They stay rounded. They've just attached in the first 15 minutes after adding them. But by 4 hours they do what we call it intercalate, which means they're part of the endothelial monolayer. The endothelial cells are pushed apart, and the cancer cell sits there in the between the endothelial cells.
Do they attach exactly at the boundary between two cells, or it's random attachment?
Okay, so it's random, but then they, so I haven't shown this here, but then they extend protrusions to find a junction.
And
they look,
sensing for junction?
yeah, like Philopodia and the Melopodia. Oh, well, I don't think they have a sensor. I think they're just... they're just extending protrusions, and then eventually they'll push one through a junction. and then then they'll go there to that hole. Yeah. So, but I don't think they sense the junctions particularly. So then, but at 24 hours afterwards, we can see that they've gone. Where's that stick gone? It's much better than the pointer. Okay. So at 24 hours, they've gone underneath the endothelial cells, and some of them have invaded quite a long way away underneath. But at least they're underneath, and the endothelial cells we can see have resealed in this stage. So although they made a hole here and sat between two endothelial cells, now they've gone through, and the endothelial cell has resealed around the cancer cells that have gone through. So what I'm saying is that they can get through, but in the 2D system, then they can't because they're too fat and their nuclei don't deform enough. Okay.
How fast are monocytes in that system?
Oh, okay. So we've done monocytes. Actually, we've done T cells in this system, but they're very fast. So they'd be through and they'd invade a massive distance in 2 hours. So yeah. They're much faster, and they can go through the collagen gel much faster as well, and much further. Yeah. Now.
Maybe the battery's off again.
Okay, everything's gone.
No.
That's fine.
Yes,
Okay, no, it's fine. I'll use my manual situation. So, I have to say sorry to everyone who doesn't like signal transduction, but it's great and I love it, so I'm going to tell you. So, because I was brought up with it, so you know, it's like in my genes. So that's what we study is signaling in cell migration, and of course a lot of people model signal transduction in cells. So we have lots of receptors on the surface of cells that are important for the process that I'm talking about. So integrins and cadherins of cell adhesion. So cadherin, cell adhesion, integrin can be cell or cell extracellular matrix. So they're probably the two most important to think about in this context. Okay. But the end product of all this signaling is to regulate the actin, the microtubules, and cell adhesions. So all the signaling, you can think that there's receptors on the surface, somehow they've got to affect all the things you've been hearing about the actin, and so on. So in between we have these signal transducers, which is what we mostly work on, which are activated by these cell surface receptors and act inside the cell to pass the signal from the receptors to the cytoskeleton and cell adhesions. Maybe I'm doing the wrong thing. Anyway, never mind. Okay. And our interest is mostly on this set of proteins called the Rho family of GTPases. They're enzymes, and they act on other enzymes, which are kinases, which phosphorylate proteins or lipids, and there are also actin regulators. So I think you might have heard something about these, which stimulate actin polymerization through different mechanisms. So the whole, how they all work is not important, but the fact that you have these different steps, the receptors, the signal transducers, leading to the changes that we've been talking about. But it is important to say a bit about how these Rho GTPases are regulated. So they bind to guanine nucleotides, so GDP or GTP, and this has got 3 phosphates and this has got 2 phosphates. And this extra phosphate, which is negatively charged, is very important for the signaling by the protein. There's another thing that's very important, and that is that they all get a lipid added to the protein, which allows them to insert into membranes, into lipid bilayers. So they're all located in membranes. They can also come out of membranes because there's a protein in cells called GDI which can bind to this lipid and pull the proteins out of membranes. So they can cycle between being in the complex in the cytoplasm, not on membranes, bound to the GDI, and they can then be released from the GDI and bind again to membranes. So these receptors that I've been telling you about, they take so the signal coming from outside the cell, which goes to a receptor. If the proteins are going to do anything inside the cell, then they need to be activated, and they're activated by adding this extra phosphate. But what happens is that a signal will activate what's called an exchange factor that releases GDP from the molecule, so the GDP comes off, and then GTP binds just because there's much more GTP in cells than GDP. There's 10 times more. So the probability that GTP comes on is much higher than GDP. And on the other hand, there are enzymes that turn these molecules off. So they release this phosphate. They remove the phosphate. They're called GAPs. They activate the GTPase activity, and that converts the protein back to the GDP form. So it's just this addition, this extra phosphate that makes all the difference for the cell to, for the protein to talk to and activate proteins inside the cell, which include kinases that phosphorylate other proteins and actin regulators. And this leads to cell motility. Okay. So you think it might be simple, but humans have a lot of these proteins. So they have 20 Rho GTPases, 3 of these GDIs, around 80 of these which activate the protein, around 60 of these which turn off the proteins, and many of these. So we love this, so we want to work out how it all works, but I'm not going to tell you all about all of those today. So I'll talk about the 3 main best known proteins, which are Rho, RAC and CDC42 to begin with. So these regulate the actin cytoskeleton. Rho is the one that makes the stress fibers that you've been hearing about in the previous talk. So inside the cell, you need Rho to have stress fibers. And the reason you need Rho is because it regulates both actin polymerization and myosin 2, which is decorating the stress fibers. So these are made of actin and myosin 2. So that's Rho, and then RAC and CDC42 are making protrusions at the plasma membrane driven by actin polymerization. RAC makes lamellipodia, which you find at the front of cells using lamellipodial migration. In this case, we put activated RAC in the cell, and it just makes lamellipodia all around the edge, and you get a fried egg because it spreads in all directions. CDC42 can mainly make filopodia, which are finger-like protrusions of the plasma membrane, but occasionally it can also make lamellipodia as well. So CDC42 is probably the most complicated molecule in this family. Okay. So we weren't particularly happy with only knowing about 3 of them since there are 20. So a lot of what we've been doing is to look at what all 20 members of this family do inside cells. So interestingly, from an evolutionary point of view, Rho, RAC and CDC42 are the most conserved in evolution. And a lot of the ones on this list are only found in vertebrates. Okay, so these are the 20 Rho GTPases, and what we wanted to do was to work out which were important for leukocytes to cross endothelial cells and which were important for cancer cells to cross endothelial cells, reasoning that they might be different, because I showed you from the movies that the processes are very different. So what we do in this screen, which was with T cells, which are leukocytes found in the bloodstream. So T cells, so what we do is we have a layer of endothelial cells, and we plate these on a filter which has holes in it for the T cells to cross not only the endothelial cells, but to cross into this bottom chamber. And the reason for doing this is because it's an easy way to count the ability of the cells to get across. But actually in this assay we've got several steps. We've got the T cells have to attach to the endothelial cells, they have to cross the endothelial cells, then they have to get through these small pores, holes in the filter to get into the bottom. So we're actually looking at a multi-step process. And of course, once we've done the screen, we have to work out what the row GTPs are doing. So how do we screen? Does everyone know what RNAi is? Anyone not know what RNAi is? Okay. Okay, so basically what we do is, so you have messenger RNAs in the cell that code for proteins, and what we do is we put a small RNA into the cell. So you have a messenger RNA which is made of A, G, C and T, no U. And so what we do is we put in a small RNA that's complementary to the messenger RNA, so it would be U, C, G and A, except it's longer. And when you make this makes double-stranded RNA, and the cell recognizes that as being not normal and degrades it. And so this is a normal process that actually happens. It's a normal physiological way to get rid of messenger RNAs and to regulate gene expression. But we use it artificially, so we put in a small RNA which will target specifically one mRNA, messenger RNA, and then the level of that messenger RNA will go down and we'll get less protein. So it's a good way of reducing the levels of proteins. So what we've done in this, it's called RNA interference.
Okay.
And that's the short RNA I. Okay. So it's basically a way of interfering with the expression of the RNAs, and that means you get less protein. So what we do is we use these small RNAs, small interfering RNAs, against each of these genes. So we have ones that will target specifically each of these genes and then reduce the level of protein inside the cell. So for T cells, what we did was we had all these different small interfering RNAs against row A, row B, row C, whatever, and reduce the level, and then we looked to see which ones affected the ability of the T cells to cross the endothelial cells. And you'll see from this that the strongest effect was observed with row A, but RAC2 also had an effect. The other ones, so we had two different sRNAs that targeted different regions of the messenger RNA. And this makes sure that you're really being specific. So for row A, we had the strongest phenotype in reducing the ability of the cells to cross the endothelial cells. So the question is, how is this working? So this is kind of the traditional model of how a T cell or any other leukocyte moves. So the idea would be that you have high levels of RAC1 at the front of the cell, and that would induce a lamellipodium and push the front of the cell forward. And then at the back of the cell you would have high levels of row A activity, and that would make actomyosin contractility and push the back of the cell forward. So it's like a kind of toothpaste tube at the back and an extension at the front. So here I'm showing you two movies. This movie here is normal cells that have just been treated with a control. And this is these are cells that have been treated to remove the row A using this small interfering RNA. So the control cells, well, you've seen the movie before, but they basically, some of them stop to cross, and some of them are moving very fast on the surface of the endothelial cells. So what they do is they move like crazy, and then they find somewhere where they want to cross, and they stop, and then they'll go through. So these cells are moving very fast. But when we don't have, when we reduce the levels of Rho A, you'll see the cells look completely different. For a start, they don't move very much. These cells are crawling around like crazy. These cells tend to look like this. Their nucleus is in the middle, and then they have two very thin protrusions at either end. So they're going like this. They're attaching there, they're attaching there. They've never managed to get across. Whereas a normal cell has exactly this shape and moves very quickly. So these cells don't move very quickly. You can see the very long, thin protrusions. So the important thing about this is this shows us that this protein, Rho A, inside cells is not just required to make this structure, which is called a uropod, but it's also required to make this structure, the lamellipodium, because if we don't have Rho A, we neither have this structure, so which is found at the back of these cells, hopefully you can see, and we don't have this structure, which is normally at the front of the cells. So we just have two very thin protrusions, but the cell can't cross. So from this we can conclude the cells actually can attach fine without Rho A because they're attached to the endothelium. So this isn't affecting attachment, but it is affecting polarity of the cells. So cells can't polarize and they can't move on the surface, so they can't find a place to cross the endothelium. So it's the second step that is affected, not the attachment. So the fact that we don't have either end of the cell, either the front or the back, when we reduce Rho A levels, made us think, well, where is Rho A really active in these cells? So from my previous model, Rho A, according to kind of a classical description, should be active only at the back of the cell and not the front of the cell. So we wanted to actually look at this, and to do this we use what's called a fluorescent FRET biosensor, where we are using the properties of fluorescent proteins and a process called fluorescence resonance energy transfer. I'll explain this in a minute. So Rho A, as we know, is active when it's bound to GTP. And when it's active in this molecule, which I'm showing here, so this is like a protein that we've made artificially, and the protein has two fluorescent proteins at either end, GFP, which is fluoresces green, and RFP, which fluoresces red. And when Rho A is active, bound to GTP, it's going to bind to this bit, which is a bit of a protein that Rho A normally binds to. And so it recognises this only when it's bound to GTP and not GDP. So when Rho A is active inside the cell, this bit of the protein will bind to this bit of the protein. And the upshot is that the molecule bends, and now this GFP, the green fluorescent protein, is very close to the red fluorescent protein. Now GFP, when it's excited by blue light, it will emit green light, but this RFP is activated by green light, so this, when it's close enough, will pass its energy directly to the red fluorescent protein instead of emitting it, and the red fluorescent protein will then give out a red light. So you can detect when this molecule is bent and Rho A is active by looking at fluorescence of the difference in fluorescence. We actually measure this by looking at a change in the fluorescence lifetime of this, because once this is closer to this, it has a shorter fluorescence lifetime. So it actually changes the lifetime of the fluorescence. So anyway, this is the movie that shows where Rho A is active. We've got lots of these. This is just an example. So this shows the shape of the T cell. And you can't see the endothelial cells, which are all the way around it. But its back is here, so that's the uropod, the back of the cell. This is the front of the cell, and it's extending underneath the endothelial cell. So we can tell that because we also have the image of the endothelial cells. So this is extending underneath, and this is pushing from the back. And the cell, yeah?
Are you showing total thread or ratio?
Sorry?
Are you showing total thread or ratio?
Here we're showing fluorescence lifetime of the GFP,
I see.
which is used as a measure of fret. So where it's red is where there's more Rho A activity, and where it's blue there's less Rho A activity. So the thing, what you'll see is that it's very dynamic. So so first of all, you can see that there are pulses at the back, which makes sense because the back's moving and contracting, so it moves over time and it contracts. But you can see there are also pulses of Rho A activity at the front. So particularly when this extension happens, you can see that there are pulses of red, which is the Rho A activity at the front.
Is this time scale minutes, seconds, or?
That is seconds.
Okay, so it's
Yeah, so it's every 45 seconds we take an image.
Okay.
No, that's minutes.
Yeah.
Yeah, so it's every 45. Yeah, sorry. Okay. So from this information, what we know now is when a T cell, so T cell is unpolarized when it lands on the endothelial cell, but when it attaches, it takes on this polarized distribution where it has a lamellipodium at the back, at front, and a uropod, which is contractile at the back. And we know from previous work and also from our movies of the activity of Rho A that Rho A is active at the back, and we know that it acts through a kinase called ROC to stimulate actomizing contractility. At the front, it's well known that RAC is active at the front, and it activates this complex called the ARP2/3 complex to stimulate actin polymerization. But we've also shown now that Rho A is active at the front from the biosensor, and also by the fact that if we remove Rho A, there's no front anymore, so this structure doesn't form. So what we believe is happening here is that Rho A is activating what's called a formin called MDA at the front, and that initiates the actin polymerization that's required then for RAC and the ARP2/3 complex to propagate the actin polymerization. So we need this to begin everything, and then RAC can take over and promote more actin polymerization. So without Rho A, we don't get either structure, and the cells can't cross the endothelium. But we've not just shown they can't cross the endothelium, we've worked out the mechanism for that. Yeah?
Does this process compete with the activation of RAC by Rho? So previously in one of the talks, people discussed the mutual inhibition of Rho A and RAC.
Yeah.
So now you're showing that Rho A is pretty much activating the activity.
It's not activate. I mean,
Well,
I think
yeah,
RAC isn't activated independently of Rho A, but that you need the filaments generated, the actin filaments generated by Rho A for RAC to have something to
My
work
question
on.
is that, like, do you have this competition of two opposite processes?
Oh yeah, yes. So it's possible you still that you don't have Rho A and RAC active in exactly the same place at the front of the cell.
That's
So
special.
yeah, spatial very. But there's been shown by another group, at least in fibroblasts, that there's spatial separation. So Rho A, the Rho A is active right at the front of the cell as well, and RAC just behind it. So you would still have spatial segregation. But they didn't do it in the same cell, and nor have we. So they did it by just comparing lots of cells. It's very difficult to do them both in the same cell. Okay. So one other thing we've been interested in is, so we've got all these 20 Rho GTPases, as I told you, and are there differences in expression of these between different cell types? So one thing we did is to compare leukemia cells that are derived from T cells with normal T cells to see if there were differences that might explain some of the differences between leukemia, T-cell leukemia and normal T-cells. So this is done by looking at the messenger RNA levels and comparing the messenger RNA levels in lots of leukemia cells with the primary T-cells. And there are three that are higher in the leukemia T-cells than in the primary T-cells, the normal T-cells. So that's this one, row BTB1, row B, and row U. So we've been looking at these, and I think I'm going to tell you about row U because that's an interesting difference to what I told you about row A. So what we found out about row U is if we reduce the levels of row U by exactly the same mechanism with this small interfering RNAs, that we can reduce the attachment of the T cells actually these are the leukemia cells because they have high levels of row U, to the endothelial cells. So these are images where the endothelial cells are red and the leukemia cells are green. And you can see this is just this is an adhesion assay where we're simply looking at the attachment. You can see there are less green cells here than in the control. And this means, of course, that there's less these cells cross the endothelial cells less because they don't attach as much. But it's a different way to what row A is doing. So if we're measuring the ability of the cells to cross the endothelium, of course they've got to attach first. And if we're reducing this attachment, then the total number that cross the endothelium is going to be lower as well. So I won't go through this. So what we know is that row A is critical for polarizing the cell and getting both the lamellipodium and the uropod. But now we know that attachment of the T cells and leukemia cells is mediated by this protein row U. Now we don't know yet how row U really regulates attachment, so that's something we've been looking at. It's something to do with integrins for sure, because the cells use integrins to attach to the endothelial cells. Okay. So everything I've told you so far is about T cells, but as I told you, or leukocytes, cancer cells do things quite differently. So far we know that two of these proteins, row A and row U, are important for two different steps of leukocyte attachment to endothelial cells and crossing endothelial cells. But what about cancer cells? So we used a similar screen, but in this screen we're reducing the levels of the row GTPases in exactly the same way, but in this screen what we're doing is we're adding cancer cells to endothelial cells. Now I told you everything is much slower with cancer cells than endothelial cells. So to speed up the whole process in terms of doing the screen, we looked at just the first step, which is attachment to begin with, and then carried on from there. So we're simply doing an attachment assay, measuring the ability of cancer cells to attach to endothelial cells. And again, we're reducing the expression of all 20 row GTPases using this small interfering RNAs, targeting each of the genes. And here are the results. So this is a prostate cancer cell line that I showed you in the movies before that we know can attach to endothelial cells. And the ones in red are the ones which, when we reduce their expression, we see a reduced attachment to the endothelial cells. So there are actually 8 of these. So that's quite a lot. That's interesting. So what we then did was to make movies again for those 8. And I'm showing some of the movies here. So as I showed you before, which was just this control movie, the endothelial cells, actually the cancer cells push apart the endothelial cells and make a gap in the endothelial cells so that they can sit down and spread within the endothelial cells. So first of all they attach, and then they carry out this spreading. So we already know that some of these proteins, when we reduce the levels, which include these here, row A, RAC1 and CDC42, reduce the attachment. So we're now looking at movies of these cells to see if they affect other steps as well as the attachment. Because as I told you, they have to attach, and if they don't attach, they're just going to stay rounded and not attach properly. But if they don't affect subsequent stages, then eventually they're going to be able to push the endothelial cells apart and spread. Okay, so this movie. You can watch whichever cell you like, but in this movie, so in each movie, control cells are mixed with the cells that have been treated with the RNA to reduce the levels of expression. So in each movie, the blue cells are the control cells. So you can see at the end of the movie here, for example, all the control cells, so a lot of them in this movie, have spread out and flat. And you can see that here, there's another spread cell. This is the control, so the yellow ones have also spread here. What you can see is that in this one, where row A was knocked down, some of the cells where row A is reduced are still rounded, but some of them have spread. But in this case, with CDC42 reduced, all the control cells, the blue cells, have spread, but these cells are still round on the surface of the endothelial cell. So they're not really attached, and they also can't do the second step, which is to spread between the endothelial cells and to flatten out. So there's two attachment steps really that we're talking about. So there's attachment to the endothelial cells, and then there's attachment to the extracellular matrix, which is underneath and spreading between the endothelial cells. So when you knock down CDC42, so CDC42 is a protein knocked down here, neither of those steps can happen. So they can't attach very well, but they also can't attach and spread on the extracellular matrix. So of course we quantify this from lots of movies. What we see is that the control cells can quite rapidly, it's slow compared to leukocytes, but by 6 hours, around 100%, nearly 100% of them have spread between the endothelial cells. But if we reduce the levels of CDC42, then this is really impaired. Because I have to say that RNAi, this process that I'm talking about, is not the same in every cell. So one cell will have lower levels of protein than another cell. So it's not 100% efficient. So there's a variable level of protein within the cell. Okay, so what about in vivo? Because everything I've told you about so far is about in vitro. So what happens in blood vessels in vivo with cancer cells? So when we inject, so what we're doing is we're injecting cancer cells, either control or with reduced levels of CDC42, into mice, and you inject it. It's easiest to inject into a vein in the tail, so which is quite big. And what happens is that because it's a vein, the cells go back into the heart straight away, and then they go out into the lung. So if anybody thinks about blood circulation, you go through from the veins into the heart, and then you need to get oxygen, so you go back out into the lungs. So what we do is we inject these cells, and then after 10 minutes we take the lungs out and look at what the cancer cells look like in the lungs. And we inject a dye which makes the endothelial cells look red. So these are the blood vessels in red, and you can already see that in the lungs the blood vessels are very complicated. So they're very narrow. So what usually happens is that the cancer cell gets physically trapped in the capillaries. So that's what's shown here. This is a cell that has had CDC42 levels reduced, and it's physically trapped in the blood vessels. So when you inject them, they don't all get physically trapped, but most of them do, because their nuclei can't deform, and so they get trapped. Leukocytes don't get trapped, they just keep going through because they're small and they can deform their nucleus much easier. They're designed to get through these capillaries, but cancer cells are not designed to get through capillaries and they just get stuck. So it's like a physical trapping.
You typically see single cells or multiple cells?
Okay, so yes, so we do see multiple cells, but we've injected them as single cells. So sometimes they attach to each other once they've got into the blood vessels.
So this would be a cellular aggregate, so to say. What you
That's
see.
a single cell.
This is a single cell.
This is a single cell, and this is also a single cell. Yeah. Yeah. But we do sometimes see them attached to each other, or get physically trapped behind each other. So, so this is just a physical entrapment in the blood vessels. But the important point here is that normally the control cells, they get trapped, but they start to extend protrusions along the endothelial cells. So then they're extending these protrusions, they're interacting with the endothelial cells. So that's the sort of shape you see. So in this image, we've just removed the endothelial cells so you can see the shape of the cancer cell. So it's not a round ball, which it would have been when we injected it. It's extending protrusions on the blood vessels, which means it's interacting with it, it's recognizing receptors on the surface and interacting with the surface. But in this case, where we've reduced the levels of CDC42, the cell's still physically trapped, but it doesn't extend protrusions, so it stays as a round ball. So this is an important point because at later time points, if you look later, the CDC42 depleted cells disappear faster than the control cells. Okay. So, so we needed to work out what CDC42 was doing that was affecting attachment to both the endothelial cells and to the extracellular matrix, which is important for the subsequent stage of transendothelial migration. So I'm not going to go through the whole signaling and the results, but what CDC42 does, we found out, is to regulate transcription of a protein called beta-1 integrin, so an integrin. And so it activates a transcription factor, and this transcription factor regulates the transcription of beta-1 integrin messenger RNA. So when we reduce the levels of CDC42, we have reduced levels of beta-1 integrin messenger RNA, and we also have reduced levels of beta-1 integrin protein. And this is really important for the transendothelial migration. So we've shown that beta-1 integrin, and other people have shown that beta-1 integrin is very important for this process, and that it's also known that it's very important for ultimately for metastasis. So what CDC42 is really doing is affecting the levels of an attachment receptor, which is required for attachment to the endothelial cells and to the extracellular matrix. So it's inhibiting all these steps that prevent the cells crossing the endothelium. How much time do I have?
Not maybe 20 minutes.
Sorry?
10, 15 minutes.
Okay, 10 minutes. I'll just very briefly say about another of these proteins. Okay, so of course we had 8 of these proteins that affect attachment to endothelial cells. So we started to work on another one. Well, we've been working on several of them, but the one I'm going to talk about is Rho-C. Now, Rho-C is interesting because it was the first gene to be identified as a gene that specifically regulates metastasis. So cancers have to grow and proliferate, and they also, if they move, they metastasize. So Rho-C was the first gene to be identified in as a metastasis-associated gene in melanoma, so skin cancer. So that's why we were interested in it, because nobody had ever had never worked out how Rho-C actually regulates metastasis, what step of metastasis it's regulating. So we knocked down row C by using this RNAi mechanism, so reducing the levels of row C protein inside the cells. And this is 3 movies. These are controls here, and these are row C depleted cells. And of course, at the beginning, they're all rounded, as you've seen before, and then they start to spread. So the control cells. These 2 control cells, that one spread very quickly, then that one spreads, and they both spread very quickly. But the row C knockdown cells, depleted cells, stay round on the surface until the very end, when I think one of them, that one, spreads there. So they have a defect similar to CDC42 in terms of attachment and ability to spread, but row C doesn't affect beta-1 integrin, so it's not the same mechanism. So CDC42 is regulating this attachment molecule, row C isn't doing that. This is a quantification just to show that when we reduce the levels of row C, which is shown in green, compared to the control cells in blue, that it reduces this not only attachment but the ability to spread between the endothelial cells. And in an assay where we're looking at the whole process of crossing the endothelial cells and also going through a filter, similar to what I described with the T cells, if we reduce the levels of row C, again shown in green, then we reduce the levels of crossing the endothelial cells. Now actually, when we inject these cells into the mice, we see exactly the same thing. So the blood vessels again are shown in red. This is a control cell which is spreading and extending lots of protrusions down the blood vessels. And just like with the CDC42 depleted cells, the row A depleted cells are much rounder and have very few protrusions. So it's a very similar phenotype, but we know it's not acting through the same mechanism. And actually we can quantify the cells in the mice and look at the cells, the proportion of cells that have extension. So the control cells always have lots of extensions, nearly all of them, but the cells with reduced levels of row C have very few extensions. And that's consistent over time. So if you look at 6 hours or 24 hours after injecting the cells, we see very few extensions. So it's not like the row C cells that don't have row C recover, they just can't make these protrusions. Okay. Maybe I just won't talk about that. So complicated at this time of day. I'll go to the end. So row C is known to activate 2 kinases, which I think I discussed at the beginning with row A. So row A and row C can both activate these kinases, which phosphorylate myosin. They affect the activity of myosin and hence contractility. So row C activates these 2 kinases, ROC1 and ROC2, which are very similar. And we've shown that these are also required for attachment and crossing of the endothelium and ultimately for metastasis. So the question is now is, well, what is row C doing? Because it's not regulating this beta-1 integrin that we know is required for attachment downstream of CDC42. So we think it's actually purely to do with shape and actomizing contractility, and that maybe you need the cells to have a level of contractility to cross, to attach and cross the endothelium. So that's speculation. But we think it could be purely to do with cell contractility, and in this case with row C. Okay. So thinking about cancer cells and leukocytes crossing the endothelium, what I've told you is that in the T cells, row U is required for the attachment step, row A is required for the cells to polarize and crawl on the endothelium and then subsequently to be able to cross it. So we could target either of these if we wanted to reduce leukocytes crossing the endothelium, for example in chronic inflammation. But for the cancer cells, which I've talked about, prostate cancer cells, we know that CDC42 is important and by regulating the levels of this attachment receptor beta-1 integrin, and that Rho C rather than Rho A acting through these kinases is important. So, and interestingly, in leukocytes, CD42 had no effect on any step that we could find, and Rho C wasn't even expressed in T cells. So that's kind of nice. So we could target one of these pathways in cancer cells, and we wouldn't expect to affect T cells. Okay. Maybe I won't talk about that. Just to say who did the work. So the T cell work was done by 3 people in my lab, Sarah Heisman, Parag Bhavsar and Elvira Infante. And the cancer cell work was done by Nico Raymond, Barbara Bordadagua and Ritu Garg, and we had some great collaborators as well. And this is our building here, where we work. In the winter it's very sunny. I don't know about in the summer, so there's no... and the Shard, if anybody's been to London, is just behind here. Sticks up, you know, like that. Okay, thank you.
Naive question. So, do you have a way to quantify the level of knockdown? So, for the side-by-side comparison, do you know how efficient the knockdown with this?
Okay, yes. So we normally look at the levels of the protein using a process called Western blotting. We take the cell lysates and we look at the amount of each protein. So for every row GTPase we've looked at, the level of knockdown is very efficient. I wouldn't say, but we can't say for individual cells. So we're taking a global, actually, we're looking at millions of cells in these cases. So we're not looking at individual cells. But the knockdown is over 90% in those cases. So there's still some residual protein, and we can't rule out that that's having some effect. And we also can't rule out that, I mean, presumably some cells have less than others because it's completely stochastic, really. So there will be variation. What we haven't looked at is like the distribution of the variation.
So Anne, you showed us some beautiful pattern and pulses of row A.
Yeah.
And I think it was the normal T
T
cell.
cell, yeah.
And do you see those in the normal movement of those cells, or only when they're trying to squeeze through? Or
yeah.
And they always look like these very dynamic patches that turn on and off?
Yeah. So we didn't, when we first set up the system, we did it without the endothelial cells just to check it was working, and we did see similar, but I think with the endothelial cells, the squeezing at the back is much stronger than you would see in just a cell moving on the surface. So because so I think we saw these dynamic areas at much better in the transmigration than just moving on a flat surface.
And do you also see RAC or CDC42 or any of the other GTPases have this sort of dynamic pattern?
Oh, okay, yeah. So for RAC, we didn't do it in these cells. We did it in different cells, but we also saw very dynamic RAC activity, changing rapidly over time. Yeah.
You might have already said this, but did you tamper with the texture, with the tissue, or with the environment where the cells were penetrating into? Did you make it any stiffer? Did you change the stiffness of it? Did you do anything?
No, sorry. I'd love to do that, yeah. I just need someone who's prepared to do that. Yeah, it would be great. I mean, in vivo we can't really. Well, in vivo we could. There are ways to change, yeah, but it's quite complicated. But in vitro,
does it matter? Do you know if the texture matter, if the properties of the tissue matter at all?
If the properties of the tissue matter, yes. Well, I suppose there are blood vessels in bone, so you think that would be harder. Some people have imaged cancer cells in the brain. So, oh no, I think maybe it might be in the brain. Yeah, I don't think anyone's really imaged them in the bone, but they have seen cancer cells interact in the brain. There's nothing I've seen that would say it would be different in different tissues so far. But it's quite a difficult thing to do, so not many people have done it. Yeah, I haven't seen anything that would tell me it would be different, but I think it should be. The blood vessels should be different.
So you've shown very nicely so that you have this interaction between rho A and M dia, and then RAC and R23 follow that, and then presumably in the CDC42 driven processes will be also forming that first generate parallel arrays of actin. So do you think this is kind of a general theme, so that the very first event is the creation of bundled arrays of actin? The reason why I'm asking is so that in dictyostelium, for example, when you move cyclic MP signal to the side, so the very first event you see is the appearance of microspikes, which is also very similar.
There's no reason why CDC40 through a formin shouldn't do the same thing. It's just in this situation, Rho A is clearly it is probably the most important one, because when we knock down CD42, we don't. But that's just in these T cells. So maybe in other cell types, CD42 is generating the formin.
If you knock down M dia.
Okay, well, the problem is there are several. So we did try that, but M dia 1, we knocked that down and we didn't have a strong phenotype. So whether it's M dia 2 or 3 or even another formin, M dia 1 is definitely at the front of the cell where we would expect it to be.
I asked maybe a naive question, unrelated to signal transduction. What is could be a mechanistic difference between penetration of normal T cells through the cell, making a hole, and this penetration of cancer cells through lamellipod? By lamellipod-driven mechanism is clear, it's just cloning. But penetration through, how it's driven mechanism, what happens, and what could be a mechanism of...
Okay, so when the T cell actually goes through the endothelial cell, so what we've seen is that...
it's not endocytosis.
No, it's not endocytosis, phagocytosis, So what we've seen is that the T cell extends, so there's the endothelial cell, extends protrusions down, like little protrusions, and you can see them very dynamically. So it extends quite thin protrusions. I mean, not like a filopodium, a bit fatter than that. And it extends these and pushes down on the top membrane. So here are the two membranes. It pushes down on the top membrane until the top membrane meets the bottom one. And then one of those protrusions, I guess, will be the first one to push down so the two membranes come really close. Now, for that fusion reaction, you will need, and someone else has shown that you need snares. So somehow, during this protrusion, the snares must get recruited from both sides to allow the fusion of the membranes.
Snares are recruited from, because fusion, this is fusion between plasma membranes.
And plasma membrane, so it's a
and
top...
snares are usually...
Yeah.
Snares come from the outside.
Well, snares are normally on,
Snares are
you know,
normally
the endosomes will fuse with the
snares are
plasma.
normally inside the cells, and fusion.
No, they are
happens
still
outside.
inside the cell. It's inside, it's on the inner leaflet of both sides. Yeah, and the two leaflets
So
come
it's a-
together.
it's
So
fusion
it's
of-
the same concept, except what's a bit different is most people think snares are acting on two different kinds of, like a vesicle and
Yeah,
or
and this
whatever.
is fusion of plasma membrane
yeah, well, with
itself.
plasma membrane.
But if you physically push a little bit with some non-cellular
Like a bead.
indent,
Yeah.
will you obtain formation of the same poles, or these leukocytes, in addition to just pushing, produce some signals?
Yeah, for sure, because they're engaging ICAM1, which is an attachment molecule for beta-1 integrin on the surface. So the T cell is using beta-1 integrin, the ICAM1 is on the endothelial cell, so you have to have that interaction for the whole thing to occur. So without ICAM1, it's not going to occur. So there's definitely signaling going on. But how the snares get recruited, I don't know. I mean, you're thinking maybe it's just a mechanical thing. Yeah.
Do they exchange bits of membrane? Or does the
Yes.
T cell come out with its own membrane? And
Oh no, they don't exchange. No, the T cell doesn't exchange, but the top and bottom membranes end up exchanging, of course, because the ICAM1, which is just on the top, ends up on the bottom after this process. Some of it.
but the T cell membrane just comes out with the T cell.
Yeah, yeah.
It's,
The T cell's just been in a membrane hole.
okay.
So yes, it's never. Yeah.
So, I mean, normally leukocyte adhesion to endothelial cells is very flow dependent, and your assay seems to not have a flow, or does it?
Yeah, so we're using, in this case, we use T cells that have been activated, and they don't have to have flow to attach by the integrin. So naive T cells, which haven't ever been activated or seen antigen or anything, Ronan Allen has shown that you need flow for the integrins to be pulled on to activate them. But in an activated T cell, the integrins are pre-activated.
But you're confident that the signaling mechanism in the presence and absence of flow is going to be the same?
I'm not 100% confident. I'm not confident. No, I'm not 100% confident that it's going to be the same. And we have got flow systems as well, but we just haven't. We're using them with the cancer cells actually. But yes, because there's no point in duplicating what other people do with T leukocytes and flow. But I don't. It could be different. Yeah, because there's mechanical forces for sure, and we think that's also the same for cancer cells. But we're trying to think of some way. So I was discussing with people at lunch some way to mimic flow, so we can look at signaling locally, because it's quite difficult to do all the microscopy under flow. Yeah. So if you could have a cancer cell and then just pretend it was pull it in a case in a way that it would think it was under flow, but there wasn't any flow, then that would be.
I don't think hypnosis would work on cells. Are there more questions? Okay. Yeah, if that's not the case, I guess we are now free for discussion, for working out models for all the beautiful things we've seen. And Anutiga, do you have to say something?
We can proceed.